Happy morning, students. I welcoming you all to this online revision program. For your September monthly test, we are having the fourth lesson and the fifth lesson up to DNA replication. So I have sent you the PDF of this lesson, and you have to go through that evaluation part of this. Now we will revise the evaluation and also the book insight questions, which is very important for you for your exam point of view. First, we will see in fourth chapter, principles of inheritance of variation. You see what is meant by multiple allelism. What is multiple allelism? In page number 47, it is that when three or more alleles of a gene that control a particular trait occupy the same locus on the homologous chromosome of an organism, they are called as multiple alleles, and the inheritance is called as multiple allelism. And then we move on to the human blood groups, but that means ABO blood group type. There are three types of blood group ABO, which was discovered by Carl Landsteiner, and the fourth blood group AB blood group was discovered by students of Landsteiner, von Castle and Sturley, and Bernstein discovered that the inheritance of these different types of blood group in human beings is determined by a number of multiple allelic series. So these alleles are present in the chromosome 9, which is concerned with the determination of a blood group. So how these blood groups can be designated by I and also by L. So each allele produces different types of enzymes. IA and IB can produce different types of enzymes. IA allele produces N-acetyl galactose transferase, and IB allele produces an enzyme called galactose transferase. This IA and IB are codominant to each other, and in case of IO there is no terminal transferase enzymes, therefore it is called as null alleles. IA and IB are dominant and it is dominant to IO. IA equal to IB and it is dominant to IO. So what are the various genotypes of this blood group? There are 6 different genotypes that you already know. So you have to see this table, genetic basis of human ABO blood grouping system. Next is the RH factor. Know more about RH factor. From your 11th standard itself, you are studying about RH factor. That means RHD, D antigens. So what is the genetic control of this RH factor? Fisher's and Ray's hypothesis and Wiener's hypothesis. It's very important 3-mark question. Genetic control of RH factor. Fisher's and Ray's hypothesis. So RH factors involved. Fisher says என்ன சொல்றாங்க அப்படின்னா 3 different pairs of alleles located in the 3 different loci. The loci is nothing but the place of a chromosome. Okay, well, this system is commonly called as CDE system, and there are different types of alleles that occur at 3 different loci. So if the combinations la நிறைய வரும், all the genotypes carrying the dominant D allele will produce positive RH positive, whereas double recessive allele will produce negative. It's a very easy one. Next one is the Wiener hypothesis. In Fisher's hypothesis, there are 3 different alleles, but in Wiener's hypothesis, there is 8 different alleles. R1 and R2, small r and capital R is given. If capital R is produced, positive RH positive phenotype, where double recessive genotypes are produced, is a negative phenotype. Next is the how will you prevent erythroblastosis fetalis? So erythroblastosis fetalis can be prevented by giving the anti-D antibodies to the mother at the week of 28th to 34th week of gestation. So this RH negative mother delivers RH positive child. Then anti-D antibody should be administered to the mother soon after the delivery. So it will develop a passive immunity and prevents the formation of anti-D antibodies in the mother's blood for destroying the RH fetal RBC. Next is the sex determination. In this sex determination, you have to know this heterogametic and heterogametic sex determination and homogametic sex determination. So in this, sex determination in human beings is very important evaluation questions. So the genes determining the human beings are located in two sets of chromosomes. One is allosomes, another one is the autosomes. There are 23 pairs of human chromosomes, in which 22 pairs are autosomes and one pair is a sex chromosomes. So females are homogametic, homo same. They produces XX chromosomes, whereas the males are heterogametic, producing one X and one Y chromosomes. ஸோ அதை அப்படியே எழுதி அதனுடைய கீழே இருக்கக்கூடிய அந்த பேரண்ட் கேமெட் ஃபார்மேஷனை எழுதணும். ஓகேவா? நெக்ஸ்ட் ஒன் இஸ் த ஜீனிக் பேலன்ஸ் இன் ட்ரோசோஃபில்லா. So that's a very important 5-mark question. I already discussed in the Zoom meeting. So you read that genic balance in Drosophila. Next one, what is meant by bar body and lionization? Okay, so that's also a very important question. So you look into that. And then haplodiploidy in honeybees. So how sex determination is determined in honeybees? Haplodiploidy. Haploid. So honeybees can produce two types of eggs. One is the fertilized egg, another one is the unfertilized eggs. So this fertilized eggs will produce the females, that is 2N, whereas unfertilized egg, that is 1N, produces haploid eggs. Okay, well, so this is called as haplodiploidy condition. Next one, what is meant by Hollandric genes? Hollandric genes means the genes that is present in the differential region of Y chromosome is called as a Hollandric gene. And then there are different types of inheritance of X-linked genes are there: hemophilia, color blindness. So you look into that. And then very important question is the karyotyping. So what is meant by karyotyping? In 5-mark it is asked. Karyotyping is a technique through which a complete set of chromosomes are separated from a cell and the chromosomes are arranged in pairs. So how you can prepare this karyotype? Karyotype can be prepared by culturing the lymphocytes from the human blood. So mitosis is induced, followed by the addition of colchicine to arrest the cell division at metaphase stage. And then the suitable spread of this metaphase chromosomes is photographed, and individual chromosomes are cut from the photograph and are arranged in an orderly fashion. And then applications of karyotyping is also important. So you have to read all the 5 points and write all the 5 points. Okay, next is what is pedigree analysis. You read. And then in disorders, genetic disorders, okay, Mendelian disorders are there, and chromosomal abnormalities are there. So Mendelian disorders, thalassemia, phenylketonuria, albinism, Huntington chorea. So you read all that, just go through that. Okay. And then chromosomal abnormalities, autosomal aneuploidy, that is Down syndrome and Porter syndrome, and autosomal abnormalities, Klippel-Trenaunay syndrome and Turner syndrome. அது ரெண்டையுமே படிக்கணும். ஓகேவா? ஸோ திஸ் the chromosomal abnormalitiesல ரொம்ப important question. That's all about the 4th lesson. Read all the evaluation 1 marks. Don't try to copy from the book. Okay, don't try to copy. So read and revise, memorize. Okay, then only you can able to score good marks in your public examination. Don't simply copy and submit. In 5th lesson, first molecular genetics. First you have to read the properties of genetic of a genes. Properties of genes. In page number 69 it is. So the properties of genes. So what are the various properties of genes? These genes are located in several genes are located in the same chromosomes. They are arranged in a linear fashion. They occupy specific position. They give several alternative forms. And then they may undergo sudden change. That means mutation. They are capable of having self-duplication. These are some of the properties of genes. And then what is meant by transformation? So transformation experiment of this genetic material. Okay, so adu paathukanum. அடுத்தது Hershey and Chase experiment. Chemistry of nucleic acids la. So what are the different subunits of nucleotide? Very important question. 3 units: nitrogenous base, pentose sugar and phosphate group. அது மூணையும் படிக்கணும். So nitrogenous bases pentose sugar and phosphate group. Pentose sugar போது two types of nucleic acid, DNA and RNA. DNA means deoxyribonucleic acid, RNA means ribonucleic acid. So DNA is found in eukaryotes, whereas RNA is found in prokaryotes, and the only difference between these two sugar is that there is one oxygen atom less in deoxyribose. That's an important point. And then nitrogenous bases. The bases are the nitrogen molecules having the chemical properties. So இதில் வந்து பார்த்தோன்னா properties of a base. So nitrogenous base போது there are two types of bases, purines and pyrimidines. Purine have adenine and guanine, and pyrimidine have a cytosine and thymine. And in RNA they have uracil instead of thymine in DNA. Next one is the phosphate group. They are derived from phosphoric acid. 3 active OH groups are present in this, which is helpful for strand formation. And this phosphate functional groups gives RNA and DNA the property of an acid. Okay, here the bonds are formed from the phosphate groups or the ester bonds. And then you have to read the properties of genetic material. So properties of genetic material is also an important 5-mark question. Self-replication, stability, information storage, and variation through mutation. Side headings போட்டு எழுதணும், it's very important. Okay, and then draw the structure of nucleosome. Draw the structure of nucleosome. See, this is the structure of nucleosome. You have to draw this and you have to mark the parts. Practice drawing this diagram. So questions will be asked in this. And then what is meant by nucleosome? அதனுடைய nucleosomeனா என்னன்னு explain பண்ணணும். So in this eukaryotic, the organization is much more complex. The chromatin is formed by a series of repeating units called nucleosomes. Kornberg proposed a model for this nucleosome in which 2 molecules of 4 histone particles H2A, H2B, H3 and H4 are organized to form the 8 molecules called histone octamer. So in this diagram you can able to see this. So the negatively charged DNA is wrapped over the positively charged histone octamer to form a structure called nucleosome. So very easy question, you can easily answer, read and write. Okay, and then in DNA replication. So in DNA replication you have to read the leading strand and lagging strand. What is meant by leading strand and what is meant by lagging strands? So one strand, that is the template strand with the polarity of 3' to 5', the replication is continuous and it is known as leading strand, while the other strand, that is the coding strand with the polarity of 5' to 3', replication is discontinuous, known as the lagging strand. And then what is meant by Okazaki fragments? The discontinuously synthesized fragments of the lagging strand is called as Okazaki fragments, which are joined by the enzyme called DNA ligases. So up to this you read. Okay, if you have any doubt, feel free to ask me. Thank you.